So good afternoon. Welcome to the final lecture of the day. So it is a great honor and a pleasure to introduce the Overton Award of this year. Awards are a very important thing for all of us, for ISCB. It's important because they set a role model of what we are expecting from the ISMB affiliates to do in their career. Awards is always a difficult thing because you have to take decisions. But at the same time, in the case of ISCB, it's an easy thing because we have great candidates. And in this, I have to say that this year was very easy. And we quickly got to an agreement of who we should choose this year for the Overton Award. So Dana is an absolutely outstanding computational biologist. She's outstanding because she has dared to enter in completely new fields, from network biology to single cells to combining with modeling, and I guess we are going to hear about this today. It's outstanding because she dared to enter into real biomedical applications, and we have here today a number of calls for computational biologists to go in trying to solve real problems in biology and biomedicine. And it's outstanding because she has made contributions to computational science and methods that are used by others, but is always a great contribution. She works in the Department of Bioscience and Systems Biology in Columbia. She has a number of awards, including the Burroughs Wellcome Fund Career, the Innovation Award from the NIH, the NSF Career Award, and the award from the Stand Up for Cancer. She's a mathematician by training from the University of the Hebrew University in Jerusalem, with a PhD in the year 2003 from one of our distinguished members as a PhD advisor, Nils Friedman. And then she made a postdoc with George Church in Harvard, working in regulatory networks. So I'm really, this is one of the good moments during the meeting for me to be able to present her and look forward for her talk and thanks her for all her contributions to our society and to science in computational biology. So I look forward. Thank you.
So thanks very much both for that very embarrassing introduction. Now I can only disappoint, and for the award, I have to say that while you say that your decision was made, the announcement took me by complete surprise. I didn't even know I was nominated, but I was certainly happy and excited and very honored to receive this award. So how do we, where's the thing that makes this thing go forward? Isn't this the,
Just to click the mouse.
Yes, that's what I'm doing. It's not working at all.
Where's the keyboard?
All right, I'll begin talking without the visual. So, oh, I need this as well. So what I do, or what I've been thinking about, is I was trained in computer science and mathematics originally, so I look at cell and biology and molecules as little computers. And these little computers, they need to sense their environment, both extrinsic and intrinsic, and make a lot of important decisions, such as to live or die, to differentiate or not, and to what type, to respond to the environment. This computation happens at many levels, how multiple regulatory factors come together to control the expression of a single gene, or taking another step back, how a cell incorporates multiple signals in order to decide developmental fate, or taking another step back, how a population of cells in a multicellular organism come together and coordinate a decision, such as a response to a pathogen together. And what I do is I try and understand the principles and the function of these molecular networks that allow our biology to compute and perform. And the idea is to combine data, high throughput data of all types and forms, and machine learning approaches to learn these networks. And what you really need, taking it from a computational perspective, is in order to learn a function or a little computational process, we need to sample the cell in many states, lots of inputs and lots of outputs, in order to infer the function. So the key idea is to perturb and observe, and what we really need is that our basic assumption is that if we look and we see these statistical correlations in the data that we measurement, these statistical correlations somehow correspond to the underlying molecular relationships and interactions. And, we talk in biology that, there's mountains of data, a tsunami of data, but actually our situation is not that good. Typically in machine learning, we have a relatively small number of samples and variables and unlimited samples. If we want to do something, Netflix challenge, Amazon shopping cart, you can get as much data, as many samples as you possibly need. And with all the many billions of sequencing reads, if you want to look at something such as different cell states, if you want to sample, for example, how genetics influences phenotypes, and you want many individuals, then actually the number of samples we have is very small. And so while machine learning has many conferences, what makes us as computational biologists unique, and what I and many of my colleagues in this room grapple with on a daily basis, is how do we handle a situation when we have a vast number of variables, far too many variables, a very complex domain and space of possible models, and actually a very small number of samples to deal with. In an instance of gene regulation, then sometimes we could look at all the different genes in our genome and treat each gene as a different sample. But if we want to look at more cellular phenotypes, then really almost all data sets I know have a paucity of conditions, a paucity of samples where we measure cell state and its response. And we have to figure out, as people that do machine learning and computational biology, how to deal with that. And that's something I've grappled a lot with. And one of the fun things about a talk like this is I get to go into the old archive, open the closeted stuff, and dig up things and dust off that I haven't looked at in a decade. And so I want to start with something I really haven't talked about in about clearly a decade. And I think that's one of the reasons I brought this is actually two reasons. I like this paper a lot, and I do think that till today, the concept of modularity and using modularity to boost power is something that we all use quite frequently. And this was a great collaboration with two dear friends and colleagues, Eran and Aviv, who are also Overton awardees. So this is now a paper with 3 Overton awardees, and I guess I complete the set of the first author's awardees. And the basic idea is we need power. As I said, we don't have enough samples, but biology is modular. A gene doesn't perform functions in isolation, but usually you have a bunch of genes coming together to perform a response to some type of threat to the cell. So if we can organize these genes as modules, identify what genes go together to perform this common task or function, and these genes, because they have to be co-regulated and need to be regulated at the same time and at the same place to do their job, they also have a common regulatory program. And we can exploit this modularity to cover all these, to uncover programs. It gives us a lot of boost in power. And for example, this is again from that 2003 paper, looking at an old East data set, about 140 old microarrays, and we could get a really good picture of the actual entire network. Most of our modules are functionally coherent. It actually worked much better than clustering our approach, and we could actually see that we could pick up the correct regulators in many cases. And one of the things I like doing best is putting my money where my mouth is, and indeed we managed to predict and validate 3 novel regulators of different types, both transcription factors and signaling molecules from this work. And as I said, it's been used frequently. Both my co-authors, I, and the larger communities have used this concept of modularity to really give a superpower boost in learning. And the idea is, first of all, this coherence and structure, it gives you an interpretation to the data. You don't have a fuzzball network. You actually have a better organization of the network. you have many more samples to learn with. If you, let's say, have 200 samples and 50 genes in a module, now you have enough examples to actually learn a regulatory function. And by dramatically decreasing the space of possible models, you get a substantial boost in power. And that was explicitly showed recently by Josh Tenenbaum in a paper theoretically. And more recently, transfer learning approaches have allowed us to relax a little bit the rigid request that all the genes in a module share the exact same function. Another approach to really boost your power, another thing that we all do, another thing that's really enabled us to grapple with this sparsity of data is data integration, particularly combining DNA and expression. DNA genetic variation is a wonderful perturbation and wonderful way to create a variation in many samples. It gives us an anchor to causality. The actual regulatory program, the computer program for these cells is in the DNA. So it's a wonderful integration. Modeling is key. And I can give an entire talk now about how data integration empowers learning and talk about all the things that I've been doing and other people have been doing in that. But I actually want to talk about the thing that I'm most passionate about recently. And this is taking a switch away from genomics. So most of us deal with genomics, but one way to get many samples, one way to overcome this sparsity of data is look at single cells. And we can treat and imagine that each single cell, each individual cell, if we can measure them, represents a sample. There's a lot of stochasticity in biology, so we see here a lot of individual cells, and each of these individual cells have slightly different protein concentrations or gene concentrations. And by treating each of these cells as a sample in our learning, we could actually possibly learn and have a lot of single cells to try and learn how cells process signals and how biological regulation works. So the question, and so we need technologies, and the rest of my talk I'm going to talk about joint work with my partner in crime. So Gary Nolan has been a great collaborator over the years. He's made some wonderful technologies possible to actually use these single cell technologies and ask questions about biology at the single cell level. What we really need is a large number of samples, very many single cell measured, and the technologies I'm talking about allow us to measure between thousands to millions of cells. And the idea is we need multiple parameters measured simultaneously in the same cell to perform the learning. And so Gary's been a wonderful partner for that. And that switches the picture around. Now we're back in this wonderful domain where we don't have that daunting number of variables, but we have a very large number of samples, and we could really begin asking some fun questions. So the first question is, does this idea work? Many times an idea looks good on paper, this idea that we can learn from single cells and treat each cell as a sample, but does it really work? And that is another thing that I'm pulling now out of the ancient closet. This is work that I actually did here in Boston when I was a postdoc with Karen Sachs, who was here at Boston at the time, and Omar Perez, who was a postdoc in Gary's lab. And we actually did have this single cell data. We had 6,000 single cells, and we had 11 parameters, 11 phospho-signaling molecules measured simultaneously in individual cells, and it worked like a charm. Data that was collected in one afternoon, and then run through some Bayesian network learning on the computer in 5 minutes, reconstructed about 2 decades worth of hard-earned biology. We got most of the edges in the network correctly, very few incorrect edges, a few missed edges because of cyclicity, and we even managed to predict a novel, previously uncharacterized influence of ERK on AKT. Serendipitously, this connection right here is something I've been dealing with a lot in my current work on drug sensitivity and targeted therapies that target the MAPK and P13K AKT pathway, and it's a very important feedback that's responsible for a lot of resistance. So what made it work? Why did it work? Well, we needed a few things to succeed. So we tried to see, well, did we really need 6,000 cells? And we downsampled our data to 420 cells instead, and you can see a clear deterioration in our network. We only have 6 correct edges and a ton of black incorrect edges. So we really needed all that samples. And did we need single cell resolution? Did the fact that we were measuring individual cells rather than an average population matter? So again, we had so many cells, we could average 20 cells at a time, and again, create 420 average samples, and we got further deterioration. So the single cell data, even a small sample, performed much better than the average data. And therefore, we needed really both the large number of cells and the single cell resolution, and this is stuck again, to succeed. So, okay, that was 2005. Of course, that means it's work that I did in 2004. What happened? Why didn't this follow with the flood of papers? This was really exciting at the time. It was runner-up Science Paper of the Year. It pretty much gave me my faculty job, and then silence. Why the silence? So this was done using flow cytometry, using fluorescent probes, and there is very high fluorescent overlaps. This is about some of the best fluorescent colors. This is like almost minimum overlap that you can get for only 8 colors. And if you're looking at signaling, you have different orders of magnitude and quantities. And let's say you have 2 important molecules in your network, and they're different orders of magnitude expressed differently. And let's say this overlap right here is only 1%, and as you can see, it's much more than 1%, then the bleed-in from one channel to another completely obliterates one of the signal. And many of the subsequent data sets that we collected with flow cytometry to try and really understand signaling suffered deeply from this bleed-in between channels. You couldn't really get it quantitative. You couldn't really look at function as deep as I wanted. And so, again, when you're stuck, when things don't work, you have to go back to the technological bench. And here again, I had a wonderful colleague with Gary Nolan, who really pushed the technology relentlessly, and Sean Bendel, who really made this thing happen and developed mass cytometry, which is really a game changer. And it starts like flow cytometry. You have single cells. You basically fix them to preserve the cellular state. You poke holes in them so you can flow the antibodies into the cells. And these antibodies, instead of being labeled by fluorescent probes, the big trick is you label them by heavy metal isotopes. And now these things can be measured by a device developed by Scott Tanner in Toronto. They get marched cell by cell and thrown into a time-of-flight mass spectrometer. And then these cells, basically, what you measure is a cloud of these heavy metal ions for each individual cell. And you get a profile which represents the abundances of up to 45 parameters in these individual cells. So what this technology gives us is up to 45 parameters: surface proteins, phosphoproteins, signaling proteins, transcription factors, anything you have a good antibody for, simultaneously. And this thing runs at 500 cells a second. So you can collect data sets with millions of cells. Finally, a real candy land of data for someone that wants a lot of samples. And this is a real game changer. It suddenly allowed us to ask a whole bunch of new questions, which we couldn't even dream up before. And what I want to share with you with the rest of my time is a few of these things that we've done. So first thing we wanted to ask before we dive back into network is we wanted to understand the structure of the single cell data. When we did that previous science work, we focused on naive CD4+ cells. That means we took all our single cells, and through a process of gating, we went step by step and took a narrow range of values in our cells and chucked most of the data out, ending up with a very small homogeneous subset of a certain predefined, well-known cell type. And we wanted to ask, well, now that we have the single cell technology, what is the diversity of cell types in our body? Can we take a whole tissue, for example, bone marrow, and understand all the diversity that's going on in that sample without using predefined gating? And the problem is that we have lots of cells and in high dimensions, and it's very hard for us to think in 30 dimensions and 12 dimensions and 45 dimensions. So we need something that allows us to grapple with this data, that allows us to organize and visualize this data. So what assumptions are we willing to make? In biology, it's all about assumptions. Now, there's some assumptions that would make our computation very easy. For example, if we can assume that the relations in the data are linear, PCA, a piece of cake, that's great, but we can't assume that. Is the data Gaussian? Again, normal distributions, wonderful for any computational biologist, but the data is not Gaussian. So all these two wonderful things that'll make our life easy, we're not getting that. And we want to make as little assumptions as possible. So here we sort of borrowed a tool that was developed by Mateen and Hinton, TSNA, a nonlinear dimensionality reduction method. And to give you an idea of how it works, you have this curve, let's say this high dimensional curve twisting a data, and let's assume that every single point here is a cell. And we want to map 3D into 2D so that the shape and the global shape and local distances are preserved. So you can see that it reconstructs that it's a one-dimensional curve, it reconstructs the places where the curvature is higher, and it preserves local neighborhoods. Cells that were nearby in the high-dimensional space become nearby in the low-dimensional space. So how does this work for biology? This thing is not working that well. So again, every dot here is an individual cell, and this is whole bone marrow mapped. So every single dot is an individual cell. The algorithm did not know that this was bone marrow, did not know what B cells and T cells were. We used labeling that was done independently by our expert biologists, and this literally mapped the different known subsets, each in its own separate area, quite accurately. In fact, all the mistakes are actually mistakes by the human. We checked them one by one, every single one of them. And what we're looking here is a map that allows us to visualize and look at high-dimensional data in 2D in a way that preserves much of the structure in a local neighborhood, so we can understand what the structure of this high-dimensional data looks like. The method is incredibly robust. I strongly believe in robust methods. So we could take out one marker. We could take out the 4 canonical markers that define the different immune subsets and still get something that looks remarkably similar. We can do this on different individuals, measured on different days, and still get the same canonical shape. Now, did we really need to make this nonlinearity? I mean, this computation is hard. It's slow. It took us a lot of time. So here's PCA, that's the linearity assumption. You can see that the data is clearly not linear. It all gets mushed together. You can't distinguish between T cell types. The monocytes and the B cells are all squished together. So we clearly needed the nonlinear assumption. It's also the data is very non-Gaussian. But the real thing that we could do is reconstructing what we know is always reassuring, but we can now use this to explore domains that we don't know. We can now take a tissue or a sample, such as a cancer sample, where we don't know what it's supposed to look like. And what we discovered is that cancer is clearly deformed, while all healthy bone marrows gave the exact same map, cancer gave a very different map, and a very different map for each individual cancer. Each individual cancer, what we see here is a landscape mark of the cell densities, and we can see these big, nice graphic art pictures that each cancer represents. Again, by coloring the cells based on their marker intensities, for instance, here's CD34, a developmental marker, we could begin to see what these different regions in the map represent and how the different cancer is structured. And we can use it to study something such as disease and relapse. This is diagnosed and relapse, and we could see how the cancer responded and changed over the course of before and after medicine. And looking at the markers again, we could see that the cancer became a lot more immature, a lot more CD34 stem cell-like marker combined with mature markers in different subsets and combinations. We could also use this in a real practical way, and this is a collaboration I now have with Harvard. It can actually identify very small, distinctly different subsets. So this is this is a real clinical sample of a person that had cancer, was in remission, and all these pink cells are healthy cells, and this right here is a small cluster of a small number, 1 in 10,000 cancer cells that have come back in the patient. And we could pick this up very, very clearly. Here's a more realistic clinical application where we have a bunch of normals, and we have red the suspect sample, and you can clearly see this aberrant little population here. Now, detecting minimal residual disease is something that you need an expert pathologist. Most oncologists can't do it, but here we all saw this thing ourselves, so definitely any expert oncologist can do this, not needing this pathologist. So immediate application right here. And now we're actually using this further. Jacob Levine in my lab is using this to study tumor heterogeneity. Here we have we've identified and validated subsequently with Ben Neal in Toronto tumor-initiating cell and ovarian cancer. Here we have this drug-resistant proliferating population in melanoma treated with BRAF inhibitor. So we could really use this to find these small, nasty subpopulations in cancer. Now switching to something else, can we find even more refined structure, something going beyond these big blobs? We saw these gradients in some of the pictures that I showed. So this was something that was done with Elad David Amir, who also did VSNet, and 2 postdocs from the Nolan Lab, Sean and Kara. And it was a truly wonderful collaboration between computational biologists and biologists. Even though it was across the country, they talked and Skyped and emailed a few times a day. And what we really want to know is, while B cells are relatively characterized, we don't know some basic things. For instance, we don't know even where the key process that defines B cells, that recombines the DNA for the BCR receptor, when that occurs. So we'd like to understand where the different key events during B cell development occurs. And our basic assumption, again, assumptions are critical, that development is gradual. Cells move from one cell type to another gradually. The other assumption we're making is that our immune cell system is in constant replenishment, even as adult. So if I take a single sample of bone marrow from my body, even though it would be very painful, it would include in that single sample the dynamic process of all the cells between the stem cell and immature B cell ready to leave the marrow, and all the transitional populations in between. And so rather than taking gating, that process I showed you before, and if you take the textbook and gate out all the different known population, these little tight things here, and see what's left over, 60% of the cells aren't identified in any gate. So 60% of the cells in this B cell sample aren't characterized at all. We want to switch it around, and we want to switch the question and take a continuous approach and treat development as a gradual continuum and find this line of how mature each individual cell is. So transferring, again, to a computational challenge, we want to get multidimensional single-cell data, given a bunch of multidimensional single cells. We want to order the cells one by one based on their developmental chronology. And we're going to do this assuming that all the markers in the cells gradually change. And we can actually take nearby cells or similar cells and look how they gradually change from a stem cell to an immature cell and all the intermediate steps. Now, one of the reasons that it's so great that we measured in such high dimension is now we can use this ordering, this trajectory, as a scaffold. We could use known events such as key markers, surface markers that define B cells, and now map where other events of interest such as DNA recombination, proliferation, and other important events happen along this developmental trajectory. So again, the computational challenge. What makes it hard? Again, this non-linearity, which I showed you earlier. During development, markers rise and fall, rise and fall. So if we look at only 2 markers, rather than having something linear, we have these big twist arounds. So Euclidean distance is not really a good measure, because these 2 cells, while they're very nearby in Euclidean distance, are very distant in developmental distance. Moreover, one of the prices that you pay when you have such wonderful data, when you have 200,000 B cells, is that they're not the most pristine and accurate measurements I've seen in my life. So it looks more like this, not this tight line, but these cells that almost mush into each other. And so finding this curve in this data becomes a challenge. And the way I represent single cells, most of our projects in our lab use this, not only this paper right here, is we actually switch geometry to a graph. We take each individual cell and treat it as a node in the graph, and connect each cell only to its most similar cells, the k nearest neighbors. And our assumption is that if you're really, really, really similar, then Euclidean distance is okay. And that's very nearby distance is reliable. And now if we want to look at longer distances, then we could look at walks on the graph. So if we want to look at the distance from the starting point to this distant point here, we can look at the shortest path, taking each individual step through reliable, small, nearest neighbor steps. But again, what's killing us in this data is the noise and these shortcuts. So we can get a shortcut from a stem cell to a mature B cell. And we tried a lot of methods to clean this up, but you can't really clean them all. If you have 200,000 cells, there's too many opportunities in too many ways and too many things that can happen that create these shortcuts. So you need something different. And my attitude is when biology throws noise at you, one of the best ways to fight noise is throw a little bit of random dice back at it. And this is a really cute little algorithm. I'm going to give you the key points of it, because this is a computational biology lecture. but I want to tell more stories. You can read all the full details in the paper. And the idea is, how are we going to get over these shortcuts with randomization? Rather than taking the hardcore deterministic k nearest neighbors, we defined a KLNN graph, where we take a big neighborhood of k, let's say 30 nearest neighbor cells, and then randomly generate a bunch of graphs with a subset of these, let's say 5 nearest neighbors. So the assumption here is that each of these shortcut would only be included in a very small number of these graphs, and therefore be averaged out when we're actually looking at the average of the entire cohort. And another way, so we are going to build this graph, and in each of these graphs we're going to look at the distance from our starting stem cell to each of the cells in our graph. And now we're also going to find waypoints. So we're going to randomly choose a couple hundred random waypoints. You can actually do it with a couple dozen. And now we're going to refine our distances with these waypoints, again, assuming that there will be a waypoint nearby, and we're going to use more nearby waypoints to really refine the distances of these these farther points from the start that you know, as you go down this path, you really lose resolution. And so each waypoint now averages in based on its distance, getting a much more accurate thing. And there's iterations and all sorts of other little details, but those are the key points. And so what happens when we apply it to real data? We had about a couple data sets with 200,000 cells measuring 19 B cell markers that were used to generate this trajectory. Again, the algorithm didn't know anything except for a random starting point that was a stem cell-like cell. And our Wanderlust algorithm pretty much automatically reconstructed the entire continuum from a hematopoietic stem cell to a naive, immature B cell ready to leave the marrow. And this path was very robust, so we did a bunch of, you could read the paper, pretty much anything you did. This is pretty much the most robust algorithm I've seen. Remove markers, change parameters, mix it up, change it up, do a bunch of different marrows, and you get the same answer, which pretty much also matches our prior knowledge. Here's an example. We have now 4 traces. Each of these traces comes from a different individual, Alice, Bob, Carol, and Dave. And you can see how for these different markers, you have a remarkably similar trajectory, how they overlay. And not only do they overlay in each one of these, but key features reproduce, specifically TDT. TDT is the enzyme that causes the actual DNA recombination in our B cells. And CD24, there's an intersection here. And remember, I told you that people didn't know when this recombination occurred. Interestingly, all this happens before the peak of CD10 and also before the peak of CD19. And previously, if you would ask any B cell biologist, they would say that CD10 and CD19, they're the earliest markers that represent B cells, and all this action is supposed to be happening after the rise of CD10. But what we suggest is it's actually, all this is happening, this TDT is active before CD10 goes up. Again, this is a prediction, and we can go back and see if the way we think, what our algorithm says, is right, and we can actually use the trajectory to define gates and go back to cells and look and see where the recombination is, and we use qPCR to see how many cells in each of these sorted populations along our trajectory are in each of the states. Have they recombined their DJ? Have they recombined their VDJ? And we see that as we predicted, the DJ recombination begins in population 3 and completes in population 4, and the VDJ recombination begins in population 4 and completes in 5. These are novel populations that haven't been characterized before, and what we've managed to do with our algorithm is predict very accurately and pinpoint where recombination, where DNA gets recombined during B cell developments in human. And this is really the value of an unbiased approach. We let the data guide us. We didn't say it should be after CD10 or CD19. We just let the algorithm sort the developmental timeline as it is. Now that we've defined these new novel subsets that haven't previously been defined, because we measured so many different layers, so many different things simultaneously, we could look at different things such as their signaling. And one of the most remarkable things we found is that population 3, where the DGA begins to happen, it exquisitely responds to IL-7 stimulation of STAT5. So when we throw IL-7 on the cells, Population 2 doesn't do anything, population 4 doesn't do anything, and population 3 pops up and induces STAT5 really strongly in 7 independent marrow after marrow. And what's particularly remarkable is here is the pie chart of all the different cells. Here's our B cells right here. Here's our early progenitor cells, this little narrow red slice here that's barely making a mark in our thing, which I blow up here. Population 3 is 7 in 10,000 cells in the marrow. And yet Wanderlust was accurate and had a fine enough resolution to systematically, 7 marrows, one after the other, pick up these 7 distinct, very rare, important cells and place them together in a reproducible fashion, really showing the power of the algorithm, both to be accurate and how important this accuracy was to pick up novel biology, also how important it was to measure so many cells. We would never pick up a population as rare as 7 in 10,000 cells if we didn't measure in the tens and hundreds of thousands. So we were really interested in this signaling rewiring. How does it happen? Why does it happen? And so we queried around, and we saw that IL-7, the receptor, continues to go up in population 4. So population 4 has the IL-7 receptor. The basal level of STAT5 is higher, so something other than IL-7, this yellow thing right here, is turning STAT5 up. STAT5 is basally up here. Maybe it's saturated, maybe it can't go up more, so we threw some phosphatase inhibitors and saw that no, STAT5 can still go up. There's something blocking it. We couldn't really figure out what exactly is going on, but it was very clear that there's very quick and very dramatic rewiring where we have ligand-dependent induction of STAT5 in population 3, and this is a small, tiny population, and right one development step from it, we have a STAT5, a ligand-independent induction of STAT5. Again, showing this quick reconfiguration of the signaling network and how dynamic it is. And we thought this might be a checkpoint, and it did. We went into a culture, and we saw that if you inhibit this STAT5 pathway, if you block it with JAK5, the cells then get stuck. They don't move on. They don't progress in their development. So what we really discovered is a new checkpoint in B-cell development. And this is something that we would not have, that would be completely missed by other approaches. It would be masked by averaging. If we looked at averaging, it would be dismissed as outliers if we didn't have enough single cells, really showing the power and the importance of taking a lot of single cells in high dimension. And just as a quick teaser, this checkpoint is important in cancer, and it seems that this is one of the checkpoints that go wrong in ALL. So we really wanted to, so what next? This is actually one of the things that I'm most passionate about, and I want to go further. This is B-cell development. Let's do what was done for C. elegans. For C. elegans, we have a complete accurate map of every single lineage. Let's do that for human, or at least start with the bone marrow and have such a map for bone marrow and other tissues. And this can help better understanding of our disease and regeneration. The other thing I want to get back to is networks, which was what I started out with. And particularly, it was very interesting how these networks reconfigure across development. But we wanted slightly bigger subsets, working with these 7 in 10,000 cells. Squeezing enough cells out of the marrow for each of them was really hard. So we went back to T cells. And here also we looked at mice because, again, material, it's easy to measure it, but material is hard to get. And this is work by Smita Krishnaswamy, a great postdoc in my lab. Again, a close collaboration with the Nolan Lab and a bunch of great people there, Matt, Sean, Michael from here, and a bunch of other people. And again, we wanted to see how the network reconfigures during development. Here we were comparing naive to effector memory T cells. And if you look at their textbook network, they look the same. You look at the same wiring diagram. You open textbook network, it looks like the same stick figure. But functionally, these two networks are completely different. Why and how? So we measured data with a surface panel that allowed us to distinguish the different T cell subsets and signaling molecules inside. So we have these two distinctions. We have enough dimensions in each single cells to identify who are you, what type of T cells are you, and what are you doing? What is your signaling? What is the levels of your different signaling molecules inside? And being able to measure this and stim this in a single tube allowed us to throw away and not take into account tube-to-tube variation, because really we could look and directly compare these different T-cell subsets in the exact same tube that were stimulated together, treated together, and measured together. So what should an edge look like? Let's say some kinase, some phosphoprotein A, is activating phosphoprotein B. If it's high in cell A, it should be activating the B in the cell, and we expect something that looks like this. So here is single cells, A versus B, and we're expecting to see this nice, beautiful relationship. Looking at real data, none of the real data looks like that. This is actually a good example. CD3 zeta and p-SLP76 are two direct steps. One should be activating the other, and this is about as good as it gets, and this doesn't look too good. And we can try and clean it up with the best of kernel density estimation. This is heat kernel density estimation, and again, we see this blob. Not quite the XY relationship that we would hope to see in a direct link in a signaling pathway that's just been activated. So we see that XY relations are obscured all over the place, and that's because signaling is weak and stochastic. So what can we do? And the idea here, the key trick is what we want is to understand Y as a function of X. So why don't we look at it that way? Why don't we condition on X and see how X responds to Y for each value of X? And if you condition the data, that is, renormalize each slice and see what is the behavior of Y, of SLP76, under each, here I'm taking these big chunks, but we're actually doing instantaneous windows, suddenly you see this beautiful function, this beautiful sigmoid emerging, and we see that in edge after edge. So it really captures the behavior in the full dynamic range. And remember those B cells before, those 7 and 10,000 cells that are responding to tile stimulation. We have a lot of rear cells. They're very important cells. These are checkpoint cells. So this really allows us to look at the full spectrum of cells in our sample. Yeah. Now, that's a pretty picture, but we want to quantify. Again, just like the visual doesn't work, any type of mutual information or similar measure, if you sort of split it up, focuses all over this blob right here, and the mutual information between X and Y, correlation, adaptive partitioning, MIC, all your favorite joint distribution-based methods miserably fail. But if you take that same conditional trick and sample across the entire range, suddenly you can get very accurate mutual information. This is exactly what our DREAMY, our density renormalized measure of mutual information, does. And the key is we want to model the conditional distribution rather than the joint. Just to show you a clear example of where it works, here's that same edge before stimulus, before we activated the TCR, and after stimulus. And we see that there's almost no difference, neither in the R-square or the mutual information. It looks pretty much similar in the scatter plot. Just the density, both the X and the Y, move to another location, but we still have this big peak, and we can't see if the actual relationship, if the influence is stronger. But when we do DREAMY, and here I just remove the blue and take only the high density, so red means high density, all these low density blue areas I cleaned out to make it clearer to see, we can see these clear edges. And now our DREAMY shows that it's more than double in its response, and for a given level of X, for a given level of CD3 zeta, we see that SLP76 isn't just rising because we have more CDC zeta, but the influence of CDC zeta is much higher. That is because of additional cofactors that are activated, and it's not just through the increase of CD3 zeta. We actually see a much stronger influence. And this is, again, goes back to the power of having many cells. So if we look at this region right here, the interesting region where the actual response is happening, this is just 2% of the cells. Most methods would throw it out as outliers. 2% of the cells, you can't really do much. But 2% of the cells in this data is 2,000 cells. I can learn something out of 2,000 cells. I can be very confident in this relationship. There are very few data sets that have 2,000 samples. Again, the power of having many cells. And now I can really look and look at these dynamics. So I can look down the key signaling cascade from the T cell receptor down to S6 through MAP kinase signaling, and we can see the dynamics and how the signal gets turned on and gets turned off. I could see how it activates through time. So here, DREAMY picks the peak at 30 seconds. And as we go down the cascade, DREAMY picks up the peak correctly at 2 minutes and at 4 minutes. We can even see a feedback right here where this edge completely shuts off while this edge is on. And we can see this clear dynamics. If we only took one time point, we wouldn't even get the story properly. And Dreamy picks up the correct pathway order. It picks up where the signal peaks. But all these other methods, and we tried about 5, 6, they fail to do that. And why? Here we went to simulated data, and we showed that most of these methods, adaptive partitioning, MIC, and even regular correlation, they really like these tight linear relationships. Linearity, all these statistical methods that we've developed in the past love linearity, and if you have these tight minor slopes that are really linear and tight, these methods get them really well. But if you get these noisy sigmoids like we have prevalent in biology, then the typical methods fail, and this is where Dreamy picks up these beautiful sigmoids very accurately. And now we have a method to do what we really wanted and compare. And we can compare these naive cells and these effector memory cells on the same cascade in the same tube. And we see here much stronger, as you can see, there's much clearer slopes in the naive cells. The effector memory cells are sort of flat. They respond very quickly, but they're not very sensitive to the input level. So we see that there's much more faithful, there's much more information transmitted in the naive cells, and the amount of CD3 zeta really controls much more carefully the amount of SLP77 in the naive cells versus the effector memory. Here's how we represented with the Dreamy score. Purple means higher Dreamy, and you can see much stronger Dreamy here in the naive cells. And this continues down the entire cascade. So in the naive cells, the signal is transmitted more carefully, more faithfully, and for a longer duration than in the effector memory cells, really allowing us for a comparison. And what I like to do always is put my money where our mouth is. We've made a prediction that the naive cells respond much more carefully to the signal than effector memory cells. So we had an ERK knockdown mouse, and we could see the prediction. If we knock down ERK to this degree right here, how would P6 change? And based on our function, we could actually predict it would drop in naive cells from here to here, and drop in effector memory cells from here to here. And we could actually pinpoint when this happens at 3 minutes, where this signal actually is activated in the cascade. Going back to our data, we can actually see quantitatively, as we predicted, both the degree of difference and the timing. We could see that we accurately predicted the difference between the naive and the effector memory cells. And so we validated this influence in P6 on ERK. We also had a successful validation of another edge, and similar validations for different pairs of subset. Basically, we had 5 predictions, and all of them validated, demonstrating for the first time that we could pick up the difference in the strength or the amount of information transferred in an edge in a signaling network. And what's next? We're going to now use it to study cancer, passion in our lab. Particularly, this is collaboration with Bird, Bott, and Miller, and we're going to look at EMT. And this combines the 2 things I talked about together. We're going to use Wanderlust to actually look at the trajectory of cells as they develop or transform from an epithelial to mesenchymal thing. And now we can learn how the network changes along this thing. So we're going to order the cells according to Wanderlust. And now we can look at how the network rewires. And this is still preliminary work, but you could see strong rewiring in many, many of the edges. What else is next? Well, here we're very limited by our antibodies. So we want more dimensions. We don't want to be limited by antibodies. RNA-seq is the answer. Currently, it's not enough cells. It's too expensive and not accurate enough. So together with Peter Sims in Columbia, we're developing a better technology to do that. So in summary, single cells are a real powerful and fun source. We can reconstruct networks. We can actually map the shape and the subpopulations that exist in our body and our normal and cancer tissues. Wanderlust can actually get a very accurate trajectory, sort the cells according to their developmental chronology and find important and novel transitional populations. And Dreamy can characterize the signaling along these trajectories and basically really quantify how cells transmit signals and their dynamics. Of course, one of the things, I'm one of the luckiest people in the world. I have a lot of great collaborators. The work I showed you today, some of my old friends in genomics right here and some of the early work. The Nolan Lab has been a partner through this entire thing. Gary's been great to work with, as well as many of the people in the Nolan Lab, Karen, Sean, Aaron, and Kara. and a bunch of other people have been helping us with single-cell data. Bernd, now a Dolan Lab graduate, has started in the University of Zurich, and we're doing a lot together. Our funding, and of course, I'm really fortunate to have the most wonderful, fantastic lab. These are my lab members, past and present. I told you only mostly about Elad's work and Smita's work. Here are some additional people that helped, but my lab is great, and that's the best privilege that I have, and thank you all for your attention.
So we have time for questions. Please came to the microphones, and if you can take your name before asking the questions.
My name is Gaurav Chopra. I'm in the Bluestone Lab, Jeff Bluestone Lab at UCSF. It's an absolutely wonderful talk, first of all. I mean, didn't even have to say it. But the things I want to ask is, one is all of these data and maps, is this being provided as a reference or a referral database that one could go back and see where their own populations lie until this technology becomes mainstream? That's number one. And the second thing is, I think, I don't know if this is possible, but given the possibility, let's say if you can merge two different experiments in the same way as you found out distances in the nonlinear way within the same experiment, then this could also point to the possibilities of relationship between multiple diseases or multiple cancer types and so on and so forth, more towards the personalization of cancer with respect to a particular individual. A person comes into the clinic, getting this thing done, the experiment done, and a reference database basically matches up where exactly does his sample lie among this plethora of data.
So great questions. Anything that's been published, so the first two projects or three things that I described were published. That data is publicly available both from my website and Gary's website. Gary's actually working on a large number of reference data sets that I'm less involved with, and will also make them public upon publication. So basically, once the paper is published, we immediately make the data accessible in as comfortable way as possible. And yes, one of the things I'm working on now, or one of the main things, is exactly with tumor patients. So I'm collaborating with Gary as well as multiple groups in MD Anderson to actually characterize patients. We are in the process of writing a manuscript that basically describes the landscape of phenotypes in AML. And there clearly is a landscape where each AML has their own unique combination of phenotypes, but these phenotypes do repeat against different AML patients, and we're expanding in that, and that's exactly what we're doing.
Hi, I'm Melissa Klein, UC Santa Cruz. So in DREAMY, I love the idea of modeling Y as conditioned on X. Is it limited to situations when you already know that there's a connection between X and Y?
So in the first step we tried, I always try and make things easy on myself, the first step. So in the B cell process, we took a linear process without branches. In DREAMY, we took a no network. We're now expanding DREAMY, and particularly in this cancer EMT work, where we don't know the edges and we're trying to learn them. And we're using DREAMY as an input for Bayesian network learning, and we've actually developed some higher dimensional versions of DREAMY and some scoring mechanisms to actually do Bayesian network learning, score-driven, as we all know it, but with DREAMY as its engine.
Hi, Robert Ness from Purdue. So using Bayesian networks as a multivariate model applied to snapshot data, but snapshot data taken from a highly dynamic system. Can you comment a bit about trying to model system dynamics from snapshot data in this context?
So in the past, I didn't worry about the dynamics because we had such a paucity of data that missing the feedbacks weren't the problem. It was just getting enough data sets. In many of the data that I have right here, even though you have single snapshots, because the cells are at different time points, you actually do have dynamic data in a single time point. For instance, that EMT data I showed you with quite a long span of cells along the entire EMT trajectory, that was actually a single snapshot on a single day. So what we find is that we take a population of cells, there is huge variation in the timing for those cells, and we can actually use that variation within a single sample to actually get dynamic models.
Thank you, Dana. It is my pleasure to give you now the certificates of appreciation from ISCB and the Overton Prize Medal. Congratulations.
Thank you very, very much.
Look at the picture. You want to look at the picture? And we have a present from ISCB too.
Thank you very, very much.
Look at the picture. Thank you. Great.